Welcome to Unit 1, Biology. Make sure you click on the word Biology to take you to the video link. You should be able to define the characteristics of living things and apply your previous knowledge to differentiate between living and non-living things. The goal of science is to investigate and understand the natural world, to explain events in the natural world, and to use those explanations to make useful predictions. The word biology comes from bios, meaning life, and logos, meaning study. So biology is the study of life. A biologist studies the interactions of life and the environment and the problems facing life and its environment, and proposes solutions to those problems. In order for something to be considered living, it has to have a characteristic of life. The characteristics of life that you need to get memorized are: 1, they're made up of cells; 2, they have the ability to reproduce; 3, they have a universal genetic code. In order for something to be considered living, it has to have a characteristic of life. The characteristics of life that you need to get memorized are: 1, they're made up of cells; 2, they have the ability to reproduce; 3, they have a universal genetic code, that's DNA. They are capable of growth and development. 5, they're able to obtain and use materials and energy. 6, they respond to their environment. 7, they maintain a stable internal environment. 5, they're able to obtain and use materials and energy. 6, they respond to their environment. 7, they maintain a stable internal environment, which is called homeostasis, and they have the ability to adapt and change over time. An organism is anything that possesses all of the 8 characteristics of life. A species is a group of similar organisms that can breed and produce offspring. If they cannot produce viable offspring, then they will go extinct. In this picture, down below, you see a picture of a liger. A liger was made from a tiger and a lion. Now, tigers and lions won't just hook up in nature. They only hook up because they got bored in a zoo somewhere, or we forced them to. So a liger is not capable of reproducing more ligers. They are what we call sterile. Scientists are not evil like some would lead you to believe. Scientists just are curious people. They share a common goal. It's to investigate and understand the natural world, to explain events in the natural world, and use those explanations to make useful predictions. This picture is of a bunch of different species of bees. Why do we need bees? What happened if all the bees were gone? Scientific knowledge is many things. It's public, it's historic, it's replicable, it's tentative, it's probabilistic. So you'll see science in the media and the newspaper. We work on the work of past scientists to help advance the future of science. We make sure that experiments are repeatable and we get the same results. And it's tentative, it changes all the time. That's one reason I like teaching science, is because it's never the same. Every couple of years there's always something new. And it's problem solving, using the scientific method. So sometimes you get frustrated doing science. Some may view science as difficult, but I promise if you do what I ask you to do in this course, it won't be difficult, just time consuming. Anytime you see something hyperlinked, make sure you go and watch the video that's associated with it. So a scientific method is an organized plan for gathering, organizing, and communicating information. Step 1. A scientist will wonder how or why something does what it does, and then they'll propose a question to whatever they're curious about. Sometimes we call this stating the problem. Then the scientist must gather information about the topic of his or her curiosity, and they need to research previous experiments and results. If you don't research those previous experiments and results, you may be reinventing the wheel, and that's a waste of time, and time is money. Then you make an educated guess or prediction of the experiment that will be performed. Next is my favorite part, conducting an experiment. An experiment is an organized process used to test the hypothesis. When we run an experiment, the scientist will change one factor and keep everything else the same. The thing that you keep the same is called a constant. You always have to have a constant in an experiment so you can compare what you changed to the thing you didn't change. Observations are made using all 5 senses, specifically sight, smell, and hearing. Data is collected from the observations made. An inference is an interpretation based on observation and prior knowledge. The experimental group is the group that is changed or altered. In this picture, can you tell which is which? A control group is the group not altered nor changed. It's the placebo effect. It's used as a standard for comparison, and it's the most common. You must have a control in your experiment. The one thing the scientist changes is called the variables. There are 2 types of variables. The independent variable is also called the manipulative variable. When you graph, this goes on the x-axis, the horizontal axis. It's the independent variable. So the way I remember it is when you get an IV in your arm, you lay down your arm horizontally for them to put the IV in. So IV goes on the horizontal axis. It's the variable that causes a change in another. It's what you are investigating. So it's the independent variable. The dependent variable is also called the responding variable. This goes on the y-axis. It's the variable that responds to the change that the scientist made. It's the results that you write in the data table. So click on this link and watch the little video. We have a problem. Does fertilizer really make plants grow? So if you predict that I predict that I vary the amounts of fertilizer a plant is exposed to, then I will have a change in growth. So what is the constant variable? What is the control? What is the independent variable? And what is the dependent variable? You should get those from that video. When I check your fill-in notes, I'll look to make sure that you watched the video and filled out those blanks. Step 5 is to analyze the data. What information do you record from your experiment? Using the data that you collected during the experiment, you must now analyze that data. There are 2 types of data. Quantitative, it has an N in it. N means numbers. So this is a controlled experiment that results in counting or measurements, numerical data. So quantitative may be used to make graphs or tables. Remember, you always have to have a chart, a table, before you have a graph. The second type is qualitative. It has an L in it, L for letters. So these are written descriptions of what scientists observe. It's also called observational data. There are 3 types of graph. A graph is a visual display of information. The first one is a line graph. It shows trends and continuous change occurring over time. On the x-axis, you plot your independent variable. 9 times out of 10, that's time. And on the y-axis, you plot your dependent variable. Line graphs are the most common graphs used in science. You will learn how to graph on the computer this semester in Biology 111. If you need extra help with this, make sure you arrange some office hours for you and I to sit down with Excel, or you to sit down with Excel and YouTube. So this graph shows inverse proportion. As the time goes down, the flow rate goes up. The graph on the right shows a direct proportion. As time goes up, temperature goes up. So we have indirect line graphs and direct line graphs. One is an inverse proportion and one is a direct proportion. Bar graphs are the second most common graphs in biology. A bar graph is information collected from counting. So in this bar graph, you have precipitation in centimeters for different cities. So we could say from this graph that Honolulu is the wettest city and Las Vegas is the driest city. That would be analyzing your data. A pie graph is used to show how some fixed quantity is broken down into parts of data. Pie graphs always equal 100%. When you're making your graphs, make sure you always give a title, you label your y-axis, you label your x-axis, and then you have an evenly spaced scale. Also make sure your graph takes up a good portion of the page. You don't want a big piece of white paper and a little bitty graph. Step 6 is to draw conclusions. Based on the problem and the data you obtain from the experiment, the scientist must analyze the data to determine if he or she will accept or reject the hypothesis. Draw conclusions about the problem that you tested, and then report findings so the other scientists can repeat the experiment. If you accept the hypothesis, you continue your research by developing a more specific hypothesis to test. If you reject the hypothesis, you use the data and conclusions drawn from the previous experiment to modify your hypothesis to something more logical and experiment the new hypothesis. Remember, there is no such thing as a bad or a wrong hypothesis. Don't ever change your hypothesis. Most of the time you can learn more from a rejected hypothesis than one that was accepted. In fact, that's how penicillin was invented. A hypothesis is a proposed prediction to be tested. It can be rejected or accepted. A theory is an explanation based on many observations supported by experimental results. It explains why things work the way they do. Can you think of some theories? The Big Bang Theory, creation theory, evolution theory, dinosaur theory, lots of different theories. So these are explanations based on many observations. Remember, science is not a belief system, so you will never hear me say, I believe that I saw a bird in a tree. You will never hear in math, I believe 1 + 1 is 2. You just don't hear those things. So you'll never hear me say I believe. I always write I think. And I want that to become a common practice for you as well. Again, science is not a belief system. The scientific law is a rule of nature. It's evidence summed up by related observations and experimental results to describe a pattern found in nature. There's no why or how, it just is. Can you think of some laws? How about the law of gravity? What goes up must come down. Newton's laws of motion. Alright, your favorite part of science, involving math. Yay! We're not going to worry about sig figs. You can worry about sig figs when you take chemistry, but you do need to know some basic conversions and some basic units. The International System of Measurement, or SI system, is based on units of 10. 90% of nations use the metric system. We're America. We think we're different, so we use the metric system. We use the English system. And sometimes we use the metric system. So this makes it complicated because we're always having to convert between the two. It allows scientific research to be understandable and communicable to scientists around the world. It's a modernized version of the metric system created by the French in 1960. And again, it's based on 10. So length is the distance between 2 points. We use a meter stick or a ruler, and the SI unit is the meter. Volume, we use a graduated cylinder and we read the meniscus. Volume is the amount of space a liquid takes up. We usually read in milliliters from the graduated cylinder, but the SI unit is the liter. Volume of a solid is the amount of space a solid takes up. We usually use a ruler and do length times width times height. Most of the time we use cubic centimeters, but the SI unit is a cubic meter. Mass is the amount of matter in an object. It's measured in grams. We get that from balance. It's different than a scale. Make sure you know that mass is different from weight. Density always stays constant. The density of water is always 1. The density of ice is less than 1. That's why it floats. The density of salt water is more than 1. That's why it sinks. Doesn't matter if I have a little bit of water or a lot of water. The density of water is still 1. Density is a ratio of mass to volume. So when you're finding density, you'll always take mass and divide it by volume. It's a direct relationship. So as mass goes up, volume goes up. As mass goes down, volume goes down. We usually use grams per cubic centimeter. And the more dense an object is, the closer to the center of Earth it is. So I like to use your body as an example. That's why your feet are on the bottom, because they're more dense. Your head has a lot of airspace in it, so it's less dense. It's at the top. Time is the interval between 2 events. We use seconds in science to measure time. There's no such thing as negative time. Temperature is the amount of average kinetic energy molecules an object has. Remember, kinetic energy is movement. So as the particles move a lot, the temperature goes up. When particles don't move very much, the temperature goes down. In science class, we use Celsius, and remember that room temperature is about 22 degrees Celsius. Water freezes at 0 degrees Celsius and boils at 100 degrees Celsius. But when we watch the weatherman or a weather bug, that's measured in Fahrenheit, and it's measured with a thermometer. If you need help figuring out how long the bird and the crayon is, or how much liquid is in the graduated cylinder, or the amount on the balance, make sure you come to lab early. The easiest way to do conversions is using the ladder method. First, you determine your starting point. Second, you count the jumps to your ending point. You move the decimal the same number of jumps in the same direction. So your starting point is kilometers. Remember, the decimal is always after the number. So in the number 4, the decimal is after it. So your starting point is kilometers, so you go up to kilo. Your ending point is meters, which is a base unit. So starting at kilo, you're going to go 1, 2, 3 jumps to the right. So put your pencil down after the number 4 and go 1, 2, 3 jumps to the right. Fill those wells in with zeros and you get 4,000 meters. If you need help with this, make sure you come to lab early or email me so we can set up a time for office hours. Again, this is something that I expect you to know. If you need help with it, seek me out. Email me at jbhsmelissaharris@gmail.com so we can set up some time to meet.
True or false regarding the 7 properties of life? Living things have order. That is true. Living things spontaneously generate. That is false. What is a conceptual scheme in science that is strongly supported, has not been found incorrect, and is based on the results of many observations? Good job. That's a scientific theory, or sometimes referred to as a principle.
True or false regarding the 7 properties of life? Living things have order. That is true. Living things spontaneously generate. That is false. What is a conceptual scheme in science that is strongly supported, has not been found incorrect, and is based on the results of many observations? Good job. That's a scientific theory, or sometimes referred to as a principle.